Welcome to the Cell Culture Dish podcast, Advancing Closed System Performance, how CPC's MicroCNX enables scalable cell and gene therapy manufacturing. In this podcast, I spoke with Troy Ostreng, Senior Product Manager, and David Burdge, Director of Cell and Gene Therapy at CPC, about the development of MicroCNX portfolio and how they're helping biopharma teams streamline closed system operations for cell and gene therapies. I want to start today by talking a little bit about CPC and their deep legacy in biologics and hospital settings. I'm curious, how has that shaped your ability to innovate for today's fast-moving cell and gene therapy space?
That's a great question. To answer that question fully, it almost helps to have a little bit of a history lesson with CPC and the biologics and advanced therapies industries. CPC was founded in 1978, so 47 years of innovation within connection technologies. The first biologic being released in 1982, so synthetic insulin. We followed that up by supporting the industry with our investment in open format connectors, largely found on single-use bags. We continued our dedication to serving the biologics industry with the release of steam through connectors that enable single-use systems to attach to permanent stainless steel systems as the biologics industry continued to evolve towards single use. 2009, we released our first aseptic connector. And in 2017, the first advanced therapies came out. In response to that, we released our first aseptic connector meant for use in the cell and gene therapy space. We have a long history of supporting life science applications between biologics, so large molecule biologics for pharmaceutical industry, as well as the cell and gene therapy industry. And leveraging that history with biologics and the roughly 35-year head start that biologics has on advanced therapies, what lessons can we transfer to cell and gene therapies as they grow at a tremendous rate? I mean, they're growing at roughly 3 to 4 times the rate when compared to biologics in their first 10 years. So how can we transition the lessons that we have learned through supporting biologics to the cell and gene therapy industry so that there isn't the need to relearn and reinvent? I believe that our experience as a supplier of connection technologies and biologics can facilitate that knowledge transfer and continue to enable the growth in cell and gene therapies.
And could you talk a little bit about some of the products that CPC has introduced for the cell and gene therapy industry?
So I think the main product that CPC has introduced within the last few years that's really targeted towards the cell and gene therapy market is our MicroCNX brand of products. And these are connection technology, sterile connectors that are really focused around small format technology and small fluid transfers. These were, when we developed them, were really specifically developed for the cell and gene therapy markets.
And I wanted to learn a little bit more about that. MicroCNX was the industry's first aseptic connector for small format tubing. I'm curious, what led to the development of this product and what has been the response by customers?
Within CPC, we follow a new product development process that's really focused on talking to our customers, talking to our end users, the people who are out there using the products day in and day out. And several years ago, we started hearing more rumblings in the market about cell and gene therapy and the growth with it, and that the current connectors weren't quite meeting what was required in that market. And so we started a pretty in-depth voice of customer or VOC campaign around really trying to understand and talk to not only our customers, but also other people in the cell and gene therapy market, in the biomanufacturing market, so that we could really understand what was needed from a sterile connector in that market. Some of the key things that we heard were, first of all, the product has to be easy to use. Our Septiquick line of products, which have been out for 15 years plus, is a really good starting point for that. So our customers were saying, look, we need a product that's easy to use and that we can put together and make that sterile connection in a short amount of time. Probably the second main thing was around the robustness. So our customers in particular, and then in a new market like cell and gene therapy, really are looking for a product that we knew was very robust in its design. And that really comes down to probably the main things are sterility. So ensuring that whatever is flowing through that line, that fluid line, does not get contaminated in any way, shape, or form through our connection process. The third main point I would say is ensuring that we are meeting the process requirements in cell and gene therapy, which are slightly different than what happens in our more traditional bio market. Again, leaning towards smaller volume transfers, leaning towards what in cell and gene therapy, the autologous type therapies are transporting much smaller volumes of fluid. We're not talking 1,000, 5,000 liters of fluid. We're talking 250 milliliters of fluid. So meeting the difference in the process requirements is probably the third development and what went into the thought process for that connector. What we really found is we really hit a home run with the product. When we launched the original MicroCNX product, we knew that there would be follow-on products to that we would continue to add to our lineup of MicroCNX. And we've had really, really good response from the market in terms of what our product does, how easy it is to use when compared to other methods by which sterile connections or sterile fluid management is handled. We've heard very, very good results from the product.
Some additional great requirements that are unique to the cell and gene therapy industry are the need for compatibility with PVC tubing, which is not common in large molecule biologics, as well as compatibility with some pretty aggressive chemicals like DMSO, the cryoprotectant. And then finally, compatibility with LN2 storage temperatures. So having a connector and a connection platform that is capable of going down to that negative 196C temperature, being brought back to room temperature post-thaw and functioning with the same robustness that Troy highlighted.
This is an area I really want to explore a little bit more in the conversation around sterility and closure, and then how do you incorporate automation? So there's a lot of discussion in the industry about the difference between a closed system and functionally closed system. Could you walk us through that distinction and explain the risks involved in not having a fully closed system?
Whether it's the bio market, whether it's cell and gene therapy, with our connectors, they are sterile connectors. So what we're looking for is to provide our customers with the ability to quickly connect two tubings together and make that a closed system. When we look at the differences between a closed system and we'll say like a functionally closed system versus a completely open system, there's some key differences. The open system allows any contaminations to get through at any point. So that's like very bare minimum, does not encompass anything that we do with sterile connectors. When we get to the differences between a closed system and a functionally closed system, those differences, those nuances are a little less. However, what it really gets down to is that the functionally closed system is inherently open and then is closed temporarily to allow sterile fluid to be passed from one location to another. 2 or 3 examples of what we refer to as a functionally closed system would be things like a biosafety cabinet or a hood, where things have to be moved into the BSC in order to make that sterile transfer. Lures would be another good example of what would be a functionally closed system. Lures inherently are open. You might have a cap over it, and then to flow fluid, you'd take the cap off. You would want to sterilize that area or sanitize where that connection is going, and then you could connect those. We would like those to be done under a BSC. And then finally, probably the third example would be like a closed system transfer device. And these are devices that make use of lures, and then they have small needles that allow the fluid to flow through. And really, what you're doing is you're having to sanitize those before you're actually doing any fluid transfer. In comparison to a closed system, which is a closed system at all times. So you're never having to open up the system. You're never having the system open at any point. That's where a couple of key technologies come into play. First of all is our sterile connectors, and that provides the system to remain closed 100% of the cycle time. The other technology would be tube welding. That's a technology where you're actually heat welding two tubing together to close a system. Those are the nuances between a truly closed system and a functionally closed system that kind of drive the sterility standpoint. A couple of things with some of the functionally closed systems like the BSC or the biosafety cabinet, for instance. There's a lot of cost that go into training these individuals, whether they're doing a BSC, whether they're doing tube welding, any of those. So training has to be way at the top of what is included in that, because if they're trying to do one of these functionally closed operations and there's a mishap, you could be losing a very valuable therapy at that point. One of these autologous therapies that are going to be going back out to treat one person, that could actually mean the difference between life and death for that person. So training is really top of mind because of the risk of contamination. And that's one of the big driving points of our sterile connectors is that we greatly reduce that risk of contamination in doing that connection, whether it's because of the simple design of the product or reducing the chance for an inadvertent mishap causing sterility issues with the fluid that's flowing through there. Our sterile connectors are really focused around that.
You've already touched on it a little bit, but can you go into a little bit more detail about how the CPC connectors support closed systems in cell and gene therapy manufacturing?
I would say it's really a takeoff of what we do in the bio market, right? David had explained earlier, we have a lot of experience in the bio market with our sterile connectors, and how we support closed systems in the bio market is not much different than what happens in cell and gene therapy, in that we are really providing a connection opportunity to flow fluid from one location to another and ensure that everything that's happening within that system remains sterile. You don't want any contaminants from the outside getting into that fluid flow path. And so that's where our connectors really are very, very valuable in what happens there. You could look at a disconnect technology in much the same way. So our connectors help to allow a user to put two fluid paths together and flow fluid. A disconnect is another way to sterilely disconnect those fluid flow paths. And in that case, what you're really looking at is to make sure that during that disconnect process, you're not getting any egress of any fluid out of the system, and you're not getting any contamination back into it through that same locations. So through a combination of connections and disconnection, we can really take care of whatever the customer needs are when it comes to that.
Yeah, another way to look at it is through the lens of Annex 1, EU regulations. In Annex 1, they refer to something known as an intrinsically sterile connection device. And what that means, what that's referring to, is sterile connectors and tube welding. Both of these solutions enable operations in a lower grade environment. Our sterile connectors enable operations that would normally take place in a grade A or grade B environment and ensure the same level of sterility in a grade C or a grade D environment. It also reduces the expertise of the staff, like Troy mentioned earlier. Right now, highly skilled staff is a capacity constraint within this industry. The ability to scale these highly trained operators that are well-versed in maintaining laminar flow and a biosafety cabinet is quickly being outpaced by future demand for these life-saving therapies. So by using sterile connectors and lowering the amount of skill that is required from your staff, you're truly increasing the accessibility to these life-saving therapies by inflating your labor pool.
That makes sense. It would be great to hear a little bit more about why the industry is shifting from tube welding to aseptic connectors like MicroCNX, and also what are the additional benefits?
Tube welding is an innovation born out of the blood banking industry, which I think is an important thing to state out loud. Yes, it is one of the two intrinsically sterile connection devices that I referenced, but it was created during a time when no other solution existed, and it was used largely to transfer blood and blood components. It later expanded into industries such as biologics and cell and gene therapies, as we're referencing today. But if we go back to the concept of transferring lessons learned from biologics and their huge head start to cell and gene therapy, biologics has largely shifted away from tube welding. And that's due to the advent of sterile connectors, such as CPC's aseptic line or MicroCNX line. Sterile connectors don't require capital investment. They make the sterile connections faster with less concern for connection quality due to a multitude of issues, such as tubing alignment, pinhole leaks in your tube weld, and even mechanical robustness or concerns around depyrogenation. Is that blade that is bonding your two sections of tubing hot enough? Is the mechanical robustness of that tube weld going to be strong enough for your applications? Both what you plan to do in your floor, but also for any surprises that arise.
That's great. I think that gives us a really good overview of the importance of closed systems. You mentioned some products in your answers to my questions, and I want to take a second just to focus on some of those products in a little bit more detail that you talked about. Could you take us through the MicroCNX, how its genderless design, valve architecture, and pinch, click, pull process reduce operator error and save time? Because I know that Troy mentioned about how important operator error can be. So I would love to hear a little bit more about that.
Another really good question and a good reason to highlight the advantages to our MicroCNX brand of products and our sterile connectors as a whole. David talked about the tube welding and how really labor intensive that is, how capital intensive that is. There's other things that go along with it. There's the capital. When you have a piece of equipment, you have to maintain that equipment. Sometimes it goes down. So there's a lot of activity that happens around tube welding and a lot of training that has to happen with any operator who is doing the tube welding. I had mentioned our VOC process earlier in the discussion. I had mentioned that one of the key things that we had heard from the market was that we need a product that is simple to use and that our operators can be trained very quickly. And so with our MicroCNX products, it is really wrapped around a 3-step connection process. And it literally is pinching to remove a cover, click the connectors together, and then you pull a membrane or a sterile barrier. An operator could literally do that as quickly as I just mentioned that process. So it's a pinch, it's a click, and it's a pull. That is very simple for an operator to remember. Once they do it the first time, they almost become experts in how those connectors work. And so by making it very simple for an operator to utilize and just keeping it to those 3 steps, you reduce the amount of any potential operator error to almost none. We can really focus on what's important to the other side of the operation and not have the operator worried about how they actually put these connectors together. That was a really key design effort that we put into the product and that we spent a lot of time. And as we were developing it, went out to the market to get that VOC back in terms of, is this really meeting your needs with what the connector is going to do?
Just to talk about a really cool innovation, the smallest connectors yet, the MicroCNX Nano and the ULT variants, they're designed to fit in freezing cassettes and withstand cryogenic temps down to negative 190 degrees Celsius. How does that enhance the cryopreservation workflows?
We're really proud of these 2 additions to the MicroCNX line. Both of them have been launched probably within the last 12 months or so. And they're really offshoots of our original MicroCNX or our MicroCNX standard product. The MicroCNX Alt, which stands for ultra low temperature, and then our MicroCNX Nano, which is really a scaled down version of the MicroCNX Alt, are two products that we launched that were really focused around what we were hearing in the cell and gene therapy market in terms of maybe not a shift, but a directionality towards having therapies that needed to be frozen down to those super low temperatures, those cryogenic temperatures, and able to sustain the cell viability during the lifetime of it. And so there were a number of things that went into the design of those. They're based on our MicroCNX standard products. However, we included some features in the MicroCNX Nano and the MicroCNX Alt that really helped to drive it towards those cryogenic applications. And that really comes around the size of them. So we put every effort into lowering the profile height of the connectors so that they could fit into what they call freezing cassettes in the market. Cassettes are metal boxes that the entire single-use assembly will be put into, and that typically would consist of the bag that holds the fluid, the cell therapy in this case, a tubing, and then our connector. And that whole cassette is frozen usually in a dewar, but also there's other types of freezing mechanisms, and they're frozen typically down to negative 150 degrees C to negative 190 degrees C. Without the use of a sterile connector that can withstand being frozen down to that temperature and then brought back up to room temperature, it makes that process super, super difficult. Again, it makes it to where the process cannot be assured to be a sterile connection if you don't have that sterile connector that's able to do that. There's a couple of keys with that. First of all, the ability to be frozen, but also there's other things that happen with that. And it comes with chemical compatibility of those two products. We enhance the materials that the products are made out of so that they can better handle the extreme chemicals that are used in those cryo temperature applications. David had mentioned a dimethyl sulfoxide previously, so DMSO as a cryopreservative that's used in the market. Again, took what the market was telling us and applied those to variants of our MicroCNX standard product to fit a little more niche need that was in the market.
Thank you so much for walking us through that. I wanted to just touch on one other product, and that is to ask what makes the MicroCNX Lure variant suitable for safe outside BSC sterile connections.
Part of the standard MicroCNX product line that we have, so we have standard hose barbs for terminations, and those can be like hose barbs that you could attach a 16th inch tubing onto. We have 3/32 inch hose barbs, 1/8 inch hose barbs. But then also we launched a line of lures, so male and female lures that can be used depending on what they're trying to attach to our connectors. Now, I had mentioned earlier when we talked about functionally sterile connections that lures are not truly sterile. So what our MicroCNX Lure product allows is for a customer to be able to attach a bag or a syringe to our connector, and we would ask them to do that in a BSC. Once that connection is made, then they can actually take that bag, that syringe, whatever they've attached to our connector, because our connector is sterile. Now they can take that outside of the hood, take that outside of the BSC, and they could store it in their standard operating room or anywhere else they wanted to. And whenever they're ready to make that connection, then they would make that connection with our, what I had referred to before as our pinch, click, pull method to ensure that whatever's in that bag or that syringe is sterilely transferred to another location. So MicroCNX Lure products enable the sterile connection and the sterile fluid transfer of products that may not normally be fully sterile.
Thank you so much for the explanation of the products. There's something else that really struck me in talking to you that I know that you do, which is how you partner with cell and gene therapy teams to match their unique requirements. Could you talk to us a little bit about that and how you use those connections to ensure that the CPC connectors support better product development?
Absolutely. I love the little play on words, the connections with our connectors. That's definitely something that we use from time to time. You have it right. It's all about the customer for CPC. You heard Troy reference earlier voice of customer, VOC, and how integral that is for us. That is really where we start. And we start gathering that voice of customer with a dedicated team of business and applications managers. These people are customer facing subject matter experts that support our customers by learning about their applications and guiding them towards the appropriate products. Now, when we talk about products, there's really two course buckets. We have released and available products, things that we have in our catalog that we promote at trade shows. But then we also have this separate bucket, and it falls within our new product introduction roadmap. Now, this is a roadmap and a pipeline of active projects that we are actively developing to meet an unmet need in the market based on VOC from those SMEs, our applications and business development managers. Within that, we can even segment it further into products intended for a catalog release, something that is available to every customer, as well as products for a custom application, something that is wholly unique for one customer and their application. Both of those types fall within our new product introduction process. And all of that facilitates making sure that we get the right product to our customers to meet the need of their specific application.
I love that. I always think that the best products are developed when suppliers and customers work together. So I think that's great. Can you tell me a little bit too about the chemical compatibility of MicroCNX, particularly, we've talked about DMSO and other cryopreservation media. How does that advance the cell and gene therapy preservation solutions?
I had mentioned we were talking about our MicroCNX Alt products and our MicroCNX Nano products, that they were really focused around the cryo needs of the cell and gene therapy market. And part of what's needed in that is when the cells are frozen to those extreme temperatures, they need some type of a protectant that allows them to do their job. And some of those protectants are pretty harsh. One of the main cryopreservatives that is used in the market is a product called dimethyl sulfoxide, or DMSO, that I had mentioned earlier. Again, very harsh on whatever you're flowing that DMSO through. It can attack that. It can cause it to break down. So one of the things that we also focused on with our MicroCNX Alt and our MicroCNX Nano products was to look at a variation in the material of construction of the product, or what the fluid that flows through the line actually touches with our products. And we searched quite a while for a material that we felt was very well suited for the cell and gene therapy markets, in particular for the cryopreservation portion of that market, that would be able to withstand those caustic, those harsh chemicals that are used in the preservation of those solutions. What we landed on was a product that is called polyphenol sulfone, or PPSU. And so our flow path for those MicroCNX Alt and MicroCNX Nano products are this PPSU material, which is not only very chemically sound and able to withstand those harsh chemicals, but it's also very durable and can withstand greater impacts at those very low temperatures. So let's say somebody is pulling a tube assembly with a bag and our connectors out of a cassette where they're transferring it and they bump it into something. You want a product that's very durable, very impact resistant. And that's another thing that the PPSU, or polyphenol sulfone, provides for us in relation to the MicroCNX products.
Great developments. I've had such a nice time chatting with you today about your developments and the cell and gene therapy space. I'm hoping to be able to get your thoughts, each of you, looking ahead. Where will CPC continue innovating to help streamline cell and gene therapy manufacturing as you look out to the future?
Innovation and where we're going in the future is a topic I'm incredibly passionate about. It encompasses a majority of my role. Our innovation philosophy at CPC is rooted in customer requirements. Through VOC consensus, we create a long-term roadmap of new products that support the rapid growth of advanced therapies. The ultimate customer in this industry is the patient, and they have highlighted a significant demand for autologous oncology therapies. There are barriers to meet this demand today, and those include capacity, speed, accessibility, and cost. We believe that process automation can significantly reduce these barriers. So taking a fresh look at our connectors, which are designed for human operations and manipulation, and reimagining them as something that is designed for robotic manipulation is a very high priority for CPC. We believe that this small change will support the industry's effort to reduce barriers and deliver more life-saving therapies to patients in need.
Yeah, David, from my perspective, from a product perspective, I think we've highlighted a number of times throughout our talk with you that we really focus on the customer. We really focus on our end users, the people who are out there using our product and make use of it through our VOC process. And as we move forward with MicroCNX technology, we'll certainly be looking to fill out what maybe little niche areas that are missing from our lineup of products, what things we're hearing from the market. And David brought up automation using robotics in cell and gene therapy to help reduce costs, to help increase quality, and to really help get those therapies to the customers, to the patients in a very efficient and easy process compared to what's happening today. And so we are really focused around looking at what we can do with our connectors that take them from what we've heard is, all right, your connectors are really awesomely designed for use by human interaction. But what they're lacking are features that a robot might interact with. So we're really focused around what those features are and what we could do to enhance our product or to launch a new version of a MicroCNX product that would be really designed around what's needed in the automation or robotics side of things.
Well, that sounds wonderful. Again, I really appreciate your time today. This was a very interesting conversation. Appreciate everything that you're doing to bring innovation to the cell and gene therapy space.
Really appreciate the opportunity to talk with you, to help you and your listeners understand a little more about what we do here at CPC and how focused we are and how passionate not only David and I are, but really our entire team is around meeting the needs of our customers through our product development process.
Thank you for joining us for this edition of the Cell Culture Dish podcast. To listen to other podcasts related to the discovery, development, and manufacture of biologics, please visit us at www.cellculturedish.com. And for downstream process topics, www.downstreamcolumn.com.